this video, let's talk about structure of bacterial cell wall and gram-positive and gram-negative bacteria. Long ago, there was a bacteriologist named Hans Christian Gram or H.C. Gram. H.C. Gram. Okay, he developed a staining technique called Gram staining technique. Gram staining technique. For what he developed this staining technique? This staining technique to distinguish between the two major classes of bacteria. Actually, he was doing some research work on bacteria. For his research related purpose, he had to separate different strains of bacteria. For that, he developed this staining technique. Okay, this staining technique, when he followed, it stained some bacteria purple and some bacteria pink. Okay, so he followed this staining technique and some bacteria become purple and some bacteria became pink in color. The bacteria which turned purple were termed gram-positive, and the bacteria which turned pink were termed gram-negative. Got it? Here, you should notice one important point, that the scientist was not aware of how the bacteria turned purple and pink. Got it? He just did this experiment and he got two different colored bacterial strains. That's all. But he was not aware of anything, the mechanism behind that. Okay. But later, the invention of electron microscope clarified that the color difference is due to the structural difference of cell wall. Got it? This color difference is due to the difference in, or the structural difference of cell wall between the gram-positive and gram-negative bacteria. Then let's see the structure of the cell wall, the general structure of cell wall. Okay, we know that the plasma membrane forms the boundary of cytoplasm. Got it? This is the plasma membrane. The last physical class we discussed it. This is the plasma membrane. Okay, the plasma membrane forms what? The boundary of the cytoplasm or protoplasm. And we can call it as a universal membrane. Why? Because its structure does not change irrespective of the type of cell. Always its structure will be same. Whatever may be the cell, whatever may be the organism, the plasma membrane is formed of lipid and protein. There is no change at all. So we can call it as a universal membrane. Universal membrane. Right? Then, actually, what lies outside the plasma membrane determines whether the bacteria is gram-positive or gram-negative, because the plasma membrane is common, no, the structure is same, but what lies outside the plasma membrane, that we know, that cell wall, and that cell wall determines whether the bacteria is gram-positive and gram-negative. Here, outside this plasma membrane, we can see this cell wall, this one. In our physical class, we discussed about the cell wall of bacteria. The bacterial cell wall is made up of peptidoglycan. You know that, okay? And outside the plasma membrane, the cell wall that is made up of peptidoglycan, it is also known as murine. Murine. Okay, murine. Murine. Okay, and in both gram-positive and gram-negative bacteria, and it is formed of sugar and proteins. It's made up of sugar and protein. We can say that it's a polymer of small subunits called NAG and NAM. That means here this particular cell wall is made up of two small subunits known as NAG and NAM. Here, this much portion forms the peptidoglycan or the cell wall. And in this cell wall, you can see small subunits arranged one after the other. They are connected by means of a small strip or a bond. So, the first one is known as NAG Second one is known as NAM. And this NAG and NAM are alternately arranged. What is the full form of NAG? I'll show you. Here, NAG is N-acetyl glucosamine. NAM is N-acetyl muramic acid. So I told you the cell wall is made up of protein and sugar. So here you see is glucose plus amine. Glucose is a sugar and amine it is a protein. So it's a protein sugar complex. NAG is N-acetyl glucosamine and NAM is N-acetyl muramic acid. These two structures are alternately arranged and form what is known as cell wall. And we can say that the cell wall is a polymer of NAG and NAM. Here M represents N-acetyl muramic acid and G represent N-acetyl glucosamine. These two are alternately arranged, you can see in this direction. Here this M and G are connected by means of another bond, and that bond is known as glycosidic bond. Glycosidic bond or interlocking sugar. Okay, so this forms the interlocking sugar, glycosidic bond. You should remember this. This is extremely important structure. Okay, it's a bond. That bond is known as glycosidic bond or interlocking sugar. Here also, in this way, different layers are arranged, not one layer. Number of layers are arranged one after the other, or we can say one above the other. Different layers are arranged. These layers are connected by means of another bond, and that bond is known as interpeptide bond. It's known as interpeptide bridge or interpeptide bond. It's also known as peptide protein cross link. Peptide protein cross link. Got it? So the interconnecting bond of these two adjacent. Then what would be the defensive mechanism of our body against this bacteria? Actually, this cell wall protects the plasma membrane, and cell wall protects the organism from external pressures. Okay, here then what would be the defensive mechanism of our body against this bacteria? You see, our body secretes certain lysozyme. That means our body secretions like tears, saliva, then sweat, etc. contain certain substances known as lysozyme. Actually, lysozyme is produced by lysosome. You know that. Okay, this lysozyme breaks interlocking sugars. This lysozyme breaks these sugars, and thereby the tears or this lysozyme or other chemical or enzymes can access what the plasma membrane, and it can attack the plasma membrane and cytoplasm, thereby the bacteria can be destructed. And one more defensive mechanism, that is external medicine like penicillin and some other antibiotics, blocks the formation of this interpeptide bond. This interpeptide bond formation will be blocked by certain antibiotic like penicillin. So these are the two important measures or defensive mechanism against the bacteria. In the next video, we will discuss gram positive and gram negative bacteria in detail. Till then, bye.